All right, so we continue on in chapter 19, and we're going to look at the breathing and respiratory movements. This is going to be slides 36 through 49. So, as we go through this portion of it, we're going to look at some various foundational things, and one of them is about how the lungs are set up. Now, we've talked about both the conducting circuit and the respiratory zones in the earlier chapters, that how the respiratory zone is really the area where gas exchange is going to happen, and how our lungs are going to have a portion of space that is the tubing system. You know, the trachea, as it comes down, and then it's going to divide into these primary bronchi, and then the primary bronchi are going to divide up into secondary bronchi, and then those are going to divide up into tertiary bronchi, and so forth. But it's not until it gets down to the little gas exchange parts, these little alveoli, that there's going to be respiration. And so we're going to look at how it's divided up into what's called dead air spaces, or dead space. And so the air that is in the tubing is considered a dead space because there's no gas exchange. So remember, the term anatomic simply means what it looks like. So this is exactly what we're talking about. Those tubes where there's no respiratory membrane for gas exchange, there's not going to be gas exchange. So it's called anatomic dead space. Now, this, under normal, in a normal healthy person, is a certain volume. And again, like we say, it's the conducting zone. Now, if we have some sort of problem with our lungs where some alveoli are not working, that is called alveolar dead space. So this is going to be abnormal. This is going to be something that is going to happen, again, from a disease process. Now, the anatomic dead space, that is normal. The alveolar is not normal. If we have that and we combine the two, we come up with what is physiologic dead space. This is the total of both of those. So, and as it says here, under normal circumstances, the anatomic dead space is equal to the physiological dead space, because there is no portion of it that is considered alveolar dead space, because under normal conditions, our lungs are functioning well. So when we're looking at this, again, we can look at the how much air enters into the lungs, and we know how large of a volume stays in that anatomic dead space. And so when we start looking at how much air actually gets into the area of respiration, we can figure out the volume of that air. So the volume of new atmospheric air moved into the respiratory passages each minute, that's going to be the tidal volume plus the respiration rate. So the tidal volume is generally, on a healthy person, 500 milliliters, right? And let's say the average person is going to breathe about 8 breaths per minute, something like that. And I'm just throwing this out there. This is not normal there. So I can figure out that if I have 8 breaths per minute, and each breath I move 500 milliliters in there, I can calculate how much air has gone through my lungs with each minute. Now, the alveolar dead space is when we take the tidal volume, and now I separate the dead space out of it, so the physiological dead space. And remember, if it's normal, this I should never start trying to write words on this. If this is normal, physiological dead space is simply anatomical dead space. So I know how much air doesn't reach those alveoli. And I just remove that from the equation. And now I can figure out how much air gets into the alveoli, and I can figure out the alveolar ventilation rate. The minute ventilation rate is how much air is entering into the system. The alveolar ventilation rate is how much air actually gets to the respiration point. So when we're breathing, we talked about inhalation, we've talked about exhalation, but there are some other non-respiratory air movements, things that happen, laughing, crying, as it says here, sneezing, coughing. So these are areas, again, which are not meant to cause gas exchange. They're meant to clear the system, kind of basically. So this is a list of some of those. Coughing, again, is going to be breathing out. It's going to clear the passageways. You ever swallow down the wrong pipe, you'll cough for a while trying to clear that passageway. Sneezing is going to be kind of clearing the upper respiratory passageway. Anything that gets in there disturbs that mucous membrane, going to cause you to try and sneeze. Laughing, again, is just, as it says, an expression of happiness, just like crying is generally an expression of sadness. Sometimes, oddly enough, crying can be an expression of happiness as well. Hiccuping, we were not really sure what it is. Most people think it has something to do with an imbalance of the diaphragm where it'll kind of twitch. Yawning, again, not something that we're really sure about what it is. We think that it is about a low oxygen content and trying to take a deep breath. And then speech, again, this is for speech. Hopefully all of those make sense. So the control of breathing is an involuntary act, right? But we can control it voluntary. So most of the time you don't even think about breathing. And it's odd that when you do think about breathing, it can really mess up your breathing pattern. So while it is involuntary, we have ability to override and control it voluntarily. Now this area is new. Again, this is new research that they've changed this, but there are 3 areas that you'll need to know, 3 specific areas, and they're going to be found in 2 general areas. The first one is going to be the medulla oblongata down here. All right, so right at the base of the brainstem, you have this medulla oblongata, and it has what is called the medullary respiratory center. And it's going to contain 2 groups, one up front called the ventral group and one in the back called the dorsal group. And then you're going to have a group that's in the pons here, and that is called the pontine group. Now, the pontine group simply controls the upper limit. So we talked about of air coming into the lungs. So we talked about how total lung volume was 6,000 milliliters, but in our little picture, it doesn't get all the way up there. So we listed at 5,800 milliliters, because if we tried to get all the way up to 6,000, we have a chance of rupturing those small little simple squamous cells. So our body caps it at 5,800. Well, the pons is the thing that controls that cap, and we're going to look at a reflex that it uses. So you can remember, because the pons is the upper portion of this, the pons is the, compared to these two, is the top portion, so it controls the upper limit of air that can enter the lungs. Now the ventral and dorsal group we're going to talk about a little more on the next one here. So this little flow chart, the pontine group is setting the upper limit. That's why it's here. The ventral group is controlling normal rhythm of breathing, the basic rhythm of breathing. And so right up front, you've got a maestro kind of, you know anything about an orchestra, you've got this guy with the back to the audience, and he is controlling all the music. The ventral respiratory group is controlling the music. Now, if you've got something that, if you've ever heard that there's, pay no attention to the man behind the screen, because a lot of times there are people in places that are behind the scenes that control things. Well, the dorsal respiratory group is behind the scenes. Dorsal means posterior or back. And behind the scenes, this little area is receiving all kinds of information, and it helps to modify the normal breathing when needed. All right, so it's behind the scenes, and it's deciding if I need to sneeze or cough or whatever. And so it's trying to help, control the normal breathing. But the basic rhythm of breathing is up front on the ventral group. The control, getting all kinds of information from different parts of the body that help regulate that, in the dorsal group. And then last and not least is the pontine group, which is just simply there to control the duration and the amount of inspiration. Now, before we get to how our body's going to help modify that dorsal group, we have to talk a little bit about what it is measuring. And so we're measuring gas, the portion of gas in our body, in our blood particularly. So we have two gases. We have oxygen and we have carbon dioxide. And both of them are vital. One of them is a waste product, carbon dioxide, and the other one is what's needed to be taken to the cells, and that's oxygen. Right? So our body measures these, and the way it measures them is what's called partial pressure. And this is another type of chemistry thing that's a measurement of the mixture of each gas in a mixture. And the way gases work, and this is for all gases, but we're just concentrating on these two, right? The way gases work is if I have, like, let's say I've got a balloon here, and I put all kinds of different air into it, and in here I've got multiple types of gases, carbon dioxide, and let's just say nitrogen. We've got all kinds of different gases in here. I'll just leave it at this. And I know the total pressure, let's say, is 10 atmospheres of pressure. That's a measurement. And I know that what I can do is, if I measure how much of each gas is in there, and I know certain coefficients, I can figure out the pressure of each gas, because each gas acts as its alone. And so the total pressure is the partial pressures of each of the gases. So if I know that oxygen has a partial pressure in this of 5, carbon dioxide has a partial pressure of 3, I automatically know that nitrogen's got a partial pressure of 2. So partial pressures are just looking at the concentration of each gas in a way of percentage in a mixture. And that's all a partial pressure is. And so it's listed as this. PO2 is the partial pressure of oxygen. And so when we start looking at the next section, we're going to talk about how our body's measuring the partial pressure of oxygen and carbon dioxide at different areas. And because that is what's going to affect our breathing. As you can see, the first two here, partial pressure of these two affect breathing. I need to keep a good amount of oxygen in my body, because if I don't, I'm going to start needing to breathe heavier to get more oxygen in, because oxygen is what drives my machinery. Partial pressure of carbon dioxide. Carbon dioxide is a waste product, but it's needed in our body as well. So I have to keep it at a certain level. And if it gets too high, I need to start breathing heavier to get more CO2 out. If it gets low, I need to shallow breathe to conserve more. Now, we're going to learn that, and I'm going to tell you this, CO2 in our body acts as an acid. Now, we know that pH is all about hydrogen ions, and we're going to talk about how this is in a minute. Excuse me. Hey, one of those non-respiratory movements, huh? But carbon dioxide acts as an acid. So if I increase carbon dioxide, I will push my body towards acidosis. If I decrease it, I push it towards alkalosis. Now, the next one is hydrogen ions. Now, this is directly related to pH. All right, so hydrogen ions is completely related to pH. There is a degree of stretch of the lung tissues. The more it's stretched, the more it's going to affect my breathing. Emotional state's going to affect your breathing. You can kind of tell, you can hyperventilate, which just simply means breathing fast. That happens normally. For instance, it would happen if I have an increase of carbon dioxide normally, but if it happens from an emotional state, I'm going to start losing carbon dioxide, and that can be a problem. And levels of physical activity, because physical activity will start using up the oxygen that I have, and so my oxygen levels will decrease, and so I'll need to breathe heavier to combat that and bring them back up to normal. So the receptors involved include mechanoreceptors and central and peripheral chemoreceptors. So mechanoreceptors we're going to look at the end. They're measuring stretch. That's basically the stretch of the lung tissues. This will go back to that pontine group, because if I breathe in too much, I'm going to have a mechanoreceptor, a stretch reflex that goes back, and it's going to shut down my ability to breathe in anymore. But the ones we're going to look at here are these central and peripheral chemoreceptors that are monitoring these two, oxygen and carbon dioxide. All right. So main controlling factors are usually carbon dioxide and hydrogen ions. Both of them are about pH. Right? pH is one of the most difficult things in our body to keep regulated. So going along, looking at the chemoreceptors, it's called central chemoreceptors because it is inside the central nervous system, the ventral part of the medulla oblongata. And again, remember, this is where the medullary respiratory center is. So the ventral area is right in front of the ventral section of it, the one that's controlling normal breathing. And it is monitoring the pH of the brain. And so O2 does not have a big effect on the central chemoreceptors. And that's an important thing to know. What does have a big effect on it is carbon dioxide. Carbon dioxide has a big effect. Now, hydrogen ions generally don't cross the blood-brain barrier. There's the little position that kind of helps keep them out of it because the brain is really sensitive to pH changes. So when carbon dioxide crosses, it's going to change into what's called carbonic acid. Now, we've looked at this. I don't know if you remember this, but you have water plus carbon dioxide is going to turn into carbonic acid. And again, I wish it was a better way to do this instead of my little mouse pad, which is going to turn into hydrogen ions and C3O or HC3O, which is bicarbonate. Right? This is that buffer system. This is why if I increase carbon dioxide, I'm going to drive the equation this way and therefore increase hydrogen ions. And so that's what this is talking about. Carbon dioxide back here crosses the blood-brain barrier and binds to hydrogen to produce carbonic acid, which will eventually turn into acid. That's going to break down and it's going to release hydrogen ions. So as it's releasing hydrogen ions, the pH is going to decrease, or said another way, the acidity is going to increase. And so this is going to cause my body to react in a way that's going to send a signal and cause me to breathe deeper and more often to try and remove as much carbon dioxide, because then going back to this equation, if I decrease carbon dioxide, it pushes the equation this way and I start decreasing the hydrogen ions. And so chemoreceptors, this is what you need to know. Chemoreceptors are in the central nervous system and they're all about pH. And they measure the chemical, since they're chemoreceptors, they're measuring chemicals. The chemical that it is measuring is mainly carbon dioxide. The peripheral receptors, they're going to be in two places, and these should be very similar, they should strike a chord in your memory bank, because these are the same two areas our blood has receptors in it. And these chemoreceptors now, and again, the blood is the baroreceptors looking for pressure, and here we've got chemoreceptors, and these are going to be detecting both oxygen and carbon dioxide, but the main one is oxygen. So peripheral chemoreceptors are in the carotid sinus and the aortic bodies. Again, the aorta, this aorta is bringing all the blood, the oxygenated blood, out to the body. So if I want to test to see how much oxygen's in there, that's the perfect place to test it, because that's the original place. Then if I'm protecting the brain, right before it gets into the cranial cavity, I've got this little area called the carotid bodies, or the where the split of the carotid artery is. And so this is going to be the place to measure right before it gets into the brain. And so this is where we will have the peripheral chemoreceptors. Now, the peripheral chemoreceptors, like I said, are all about oxygen. central chemoreceptors, all about carbon dioxide, peripheral chemoreceptors, and I say mainly about oxygen. And so this is going to be, and it says peripheral chemoreceptors. This should say mechanoreceptors here, because what this is, the inflation reflex, or sometimes called the Herring-Brauer reflex, but the inflation reflex. As I breathe in, there are stretch receptors that are stimulated. As they get to a certain point, they're going to travel to the respiratory area that we looked at, called the dorsal respiratory zone area in the medullary respiratory center. And this will shut off my phrenic nerve and prevents overinflation of the lungs. And so this is the safeguard to keep me underneath that 58 milliliter threshold and not try to push it to 6,000 milliliters. So these are this is the chart going over some of the things that are going to affect it. Stretch of the tissues, prevents overinflation. Low plasma O2 is going to increase breathing, increased ventilation, increased respiration, since I've got low O2, I'm trying to reverse it and bring it up. And the same thing with high plasma, CO2. I need to decrease it. And so I'm going to go through here, and it's going to cause, since I've got a high CO2 content, which is leading to acidosis, I am going to breathe heavier and deeper, remove the CO2, and bring that value back down. And then finally, cerebrospinal fluid and the hydrogen ion concentration, or CO2 concentration in this area, is also going to increase breathing.